Okay, welcome back. This is lecture 3 for neuroanatomy, neurophysiology. We're going to be looking at a little bit of the neurophysiology in the brain. Now, I shared a little bit about this earlier, but there are two cells in the nervous system. There's the glia cells, and there are the neurons. The glia cells are the structures that form the myelin sheath around axons and provide protection and support. The neurons are the nerve cells, and they're responsible for conducting impulses from one part of the body to another. And the components of the neuron are, as we discussed in a previous lecture, the cell body, the dendrites, which you see here, and those receive information to conduct impulses toward the cell body. And then the axon, and that sends or conducts information away from the cell body. We're going to be looking here at this nice slide of the synapse or synaptic cleft. And that is the connection and area of communication between neurons where neurotransmitters are released. The synapse converts an electrical signal or action potential from the presynaptic neuron into a chemical signal, neurotransmitter, that is transferred to the postsynaptic neuron. And neurotransmitters are released at the synaptic cleft as a result of electrical activity or action potential. And there are two phases of action potential, and they're called depolarization. That is the initial phase, and that is an excitatory response when sodium and calcium ions flow into the cell. And then there's repolarization, and that is the inhibitory response when potassium leaves the cell or chloride enters the cell. And problems in either the structure or chemistry of the synapse interrupt normal flow of impulses and stimuli, which then contribute to symptoms commonly seen in psychiatric disorders. And neurotransmitters are the chemicals synthesized from dietary substrates that communicate information, from one cell to another. And the neurotransmitter will be released from the presynaptic neuron, you see here, cross the synapse, and then bind to a specific receptor from the synaptic cleft to the postsynaptic neuron. And then there's specific criteria that must be met for neurotransmitters. As you can see here, the neurotransmitter, as I said, will be released from the presynaptic neuron and cross the synapse, and then bind to a specific receptor on the postsynaptic neuron. The neurotransmitter must be present in the nerve terminal. Stimulation of the neuron must cause release of neurotransmitter in sufficient quantities to cause an action to occur at a postsynaptic membrane. And effects of exogenous transmitter on postsynaptic membrane must be similar to those caused by stimulation of the presynaptic neuron. And a mechanism of inactivation or metabolism of the neurotransmitter must exist. And exogenous drugs should alter the dose response curve of the neurotransmitter in a manner similar to the naturally occurring synaptic potential. And interestingly, there are more than 50 neurotransmitters in the brain that we know of. And we're going to look at some categories of neurotransmitters, such as monoamines, amino acids, cholinergics, and neuropeptides. So I want to look at some specific transmitters. We have monoamines, or biogenetic amines. First is dopamine, probably well known to most of you. It's a catecholamine produced in the substantia nigra and the ventral tegmental area. Its precursor is tyrosine, and it's removed from the synaptic cleft by the monoxidase enzymatic action. And there are 4 dopaminergic pathways: the mesocortical, mesolimbic, nigrostriatal, and tuberoinfundibular pathway, and we'll talk about those later in another lecture. There's norepinephrine. It's also a catecholamine. It's produced in the locus coeruleus of the pons. Its precursor is also tyrosine and is removed from the synaptic cleft and returned to storage via an active reuptake process. It's a major neurotransmitter implicated in mood, anxiety, concentration disorders. And then we have epinephrine. It's produced by the adrenal glands. The epinephrine system is also referred to as the adrenergic system. And then we have serotonin. That's produced in the Raphe nuclei of the brainstem. Its precursor is tryptophan. Remember, we've heard about that when people have Thanksgiving and eat a lot of turkey. Removed from the synaptic cleft and returned to storage via active reuptake process. It's a major neurotransmitter implicated in mood and anxiety disorders. Next, we're going to look at amino acids. There's glutamate, aspartate, GABA aminobutyric acid, and glycine. Glutamate is a universal excitatory neurotransmitter, major neurotransmitter involved in the processes of kindling, which is implicated in seizure disorders and possibly bipolar disorder, an imbalance implicated in mood disorders and schizophrenia. And then we have aspartate. It's another excitatory neurotransmitter, and it works with glutamate. And then GABA, or GABA aminobutyric acid. It's a universal inhibitory neurotransmitter, site of actions for benzodiazepines, alcohol, barbiturates, and other CNS depressants. And then glycine, that one we hear a lot about, but it's another inhibitory neurotransmitter, and it works with GABA. And then we have the cholinergics, acetylcholine. It's found in the autonomic nervous system, the sympathetic and the parasympathetic. If you remember, we discussed that in another lecture. The sympathetic, the fight or flight response. Acetylcholine is the neurotransmitter in the preganglionic neurons. And then the parasympathetic, it's the rest and digest. Acetylcholine is responsible for transmission from nerves to the organs. And interestingly, acetylcholine is synthesized by the basal nucleus of Maynard. Then we have our neuropeptides, and these have grown in study more recently over the years. You've probably heard of these. So there are some of them are non-opiate type substance, like substance P, somatostatin, and then our opiate type, the endorphins and enkephalins and dynorphins. And they help to modulate pain. A decreased amount of neuropeptides is thought to cause substance abuse. Okay, I wanted to take a few minutes to talk about the recovery and degradation of neurotransmitters. So after the neurotransmitter reaches the postsynaptic neuron, which you see here, it may then diffuse off its receptor to be destroyed, or it can be transported back to the presynaptic neuron for reuse. So here in A, you see the transmitter coming down, and then it's actually being reuptaken. And these are coming here and it's being reuptake there. Here in B, you see the neurotransmitter coming in, it's connecting, and it's being diffused off and degraded. So enzymes destroy those neurotransmitters over here. Here again, they're reuptaken.